Hi class! So today we are going to be talking about cell division, also known as mitosis. So here is a little picture of what the cells are going to look like while dividing. And of course this PowerPoint is created by me. So stay tuned so you are aware of what is going on in order to prepare yourself for the activity that we'll be doing tomorrow in class. So you may be asking yourself, what exactly is cell division? Well, I'm going to answer that cell division is a process by which a cell divides into two new cells. Now, why exactly do cells need to divide? Well, living things grow by producing more cells, not because each cell increases in size. So, I kind of hope that you are all aware that you're not created out of just 3 or 4 very large cells. You are all comprised of multiple, and I mean multiple, like hundreds of millions little cells are create your being. So it is important that cells grow and more cells are produced to create who you are. Now, skin cells create most of your body. All of the little cells that are comprised of your skin are very, very tiny. It is also important that cells divide in order to repair damaged tissue. If you happen to have a paper cut on your finger, you want that to be healed. So it is important that cells divide in order to heal that scar tissue that occurs. It, cells also divide to get enough nutrients into the cell and for waste to get out of the cell. If a cell gets too big before being able to divide, there is not going to be enough nutrients there. They can't get across the surface area of the very large cell, so they're not going to get what they need in order to survive. All right, cell division. So this is what mitosis is. And it is the original cell is called the parent cell, and that is what is going to replicate itself into two new cells called daughter cells. So it's like the mother and father creating the daughter cells. Before cell division occurs, the cell replicates or copies all of its DNA. And DNA is very, very important in cell division. So we're going to cover that a little bit after this. And each daughter cell gets a complete set of genetic information from the parent cell. And each daughter cell is exactly like the parent cell with the same number of chromosomes as the original cell, which is very important also to take into consideration. And here is the end result, which we are going to get to later in the PowerPoint. But as you can see, there are two exact replicates of what a parent cell would have been. So you have the chromosomes are the same in each cell, the centrioles are the same in each cell, there's a plasma membrane and the nuclei, and they look very, very similar to each other. And DNA, as I said, that was very important, so we're going to go over what that is. Now, DNA is located in the nucleus and controls all cell activities, including cell division. Now, the long and thread-like DNA is in a non-dividing cell is called chromatin, and chromatin looks very similar to a plate of spaghetti. And it is similar to a plate of spaghetti because it is all thin and long and they are all entangled with one another. It's very, very difficult to distinguish the beginning of a piece of chromatin and the end of a piece of chromatin specifically because they are all together in one on a plate, technically, which the plate would be the nucleus. And the double coiled short DNA in a dividing cell is called a chromosome, which is on the side. As you can see, the chromosome is the X DNA that we are very, very familiar with, and it looks like this, as you can see on the side, and it is connected in the middle by a centromere, and each individual little side is a chromatid, which I believe, yeah, here we go. So the unduplicated side is the chromatid, and then the duplicated are comprised of two chromatids called sister chromatids because they are alike, but they are also dissimilar. So they're sisters, not twins, and that is why they are together to create a chromosome. And mitosis, as I had said earlier, is the process of cell division, and is the division of the nucleus into two nuclei, and each with the same number of chromosomes. Mitosis occurs in all the somatic body cells, so the skin and the liver and all of the different organs in your body are created of cells that have specialized processes in order for you to survive. And why does mitosis occur? Is so each new daughter cell has a nucleus with a complete set of chromosomes. If certain cells are lacking the chromosomes or the necessary organelles needed to provide the specialized processes that they are created to do, then you aren't going to function properly and problems are going to occur. So the first stage of mitosis is called interphase, and is a period of cell growth and development. And this is where DNA replication occurs. Centrosomes and centrioles double, which I will cover a little later as a diagram evolves. And the cells also grow, carry out normal cell activities, and replicate all other organelles that are necessary for you to survive. And the cell spends most of its life in interphase. All right, so this is a little picture of what interphase looks like. As you can see, the chromatin looks like a plate of spaghetti, all combined together onto a little plate, which is the nucleus. And the centrosomes are up there, and you can see that there's two sets of centrosomes with two centrioles in each centrosome. And those are very important in mitosis, as we will see later. All right, so the main phases of mitosis after interphase are prophase, metaphase, anaphase, and telophase. And these are little diagrams, as you can see, that are shown and represent what occurs in each phase. So pay particular attention to that, because you will definitely be needing this knowledge for the activity that will be completed in class. So prophase, this is what happens in prophase. As you can see, the centrosomes and the centrioles created little lines, which these are called spindle fibers, and these will later be attached to the chromosomes. And the chromosomes formed is no longer spaghetti-like chromatin. They are the X-like DNA structures that we are very familiar with, and they are connected by centromeres. And it's not very well portrayed in this picture, but eventually in the end phase of prophase, the nuclear envelope disappears. So the chromosomes are kind of just floating in the plasma. And then here is metaphase. This is when the spindle fibers come out of the centrosomes and they are connected to the chromosomes that align in the middle of the cell in order to be separated, which occurs in anaphase. So the spindle fibers take a piece of a chromatid. The chromosomes are no longer connected to each other, so they're no longer chromosomes. They are individual chromatids now, and they are being taken across to the opposite poles of the cell in order to create two new cells. So this is when telophase comes into play, and the chromosomes uncoil again and create chromatin. The spindle fibers eventually disappear. And as you can see, two new nuclei are forming. So there's two new nuclei and eventually going to be two new cells. So cytokinesis is the division of the rest of the cell, which the cytoplasms and organelles after the nucleus divides. And after mitosis and cytokinesis, the cell returns to interphase to continue to grow and perform regular cell activities. So this is cytokinesis. As you can see, it's the cell, and then it's the cell with the cleavage furrow, which the cleavage furrow is what pinches together to separate the cell. And then the two new daughter cells are formed, which look exactly alike. And then in plants, mitosis also occurs in plants in order for plant cells to divide, similar to animal cells. And the same steps in animal cell division takes place in plant cell division. However, in cytokinesis, a cell plate forms instead of a cleavage furrow. So it's going to look a little different than animal cells. And this is an example because plant cells are square where animal cells are circular, and as you can see, the little cell plate formed and there's two new squares, which is two new cells. As I stated earlier, specialized cells, different cell, they live for different periods of time. So a skin cell lives for 2 weeks until they're replenished. A red blood cell lasts for 4 months. A liver cell can live for 300 to 500 days. An intestine has 4 to 5 days. And an intestine is muscle and other tissues, and those live for 16 years. So you all have actually had your same intestine cells currently since the time you were born. So they're probably about 13 or 14 years old at this point, so you have another 2 years of intestine cells, so that's pretty interesting. And the cell cycle, as I said, cells spend most of their time in interphase, so that looks like it's a good 85% of the time in interphase, and then they go to prophase, metaphase, anaphase, telophase, and then cytokinesis, which is a little more than the other phases because all the organelles and the cytoplasm have to replicate as well. And then this is what it looks like under a microscope. So as you can see, the interphase area is there's only one little nucleus with all the spaghetti chromatin in there. And then the prophase, a little more action is occurring. And then the metaphase, all the chromosomes are lined up in the middle. The anaphase, the chromatids are moving across to the opposite poles, and the telophase, the cell plate occurred, and there's 2 new cells being formed. So that's pretty neat to look at. And here is an up close and personal version of that. So it's a little easier to see, and it's very cool to look at them under the microscope. So I'm really hoping we'll be able to do that as well. And I believe that is the end of the PowerPoint, so I hope you all enjoyed and learned something, and be prepared to come to class tomorrow to make a sweet little model of some mitosis steps. So thanks for watching.